días a todas, a todos. Les doy la bienvenida a esta nueva actividad que hemos organizado conjuntamente con la Comisión Directiva de la Sociedad Argentina de Inmunología. En esta ocasión, durante el mes de junio, vamos a tener dos actividades relacionadas con un tema que es realmente apasionante, como lo es el inmunometabolismo. Quiero agradecer especialmente a Mercedes Borge, a Luciana Balboa y a Cintia Stempin, que generosamente se han dispuesto a organizar estos dos webinars con dos disertantes de lujo, como el doctor Ping-Chih Ho y el doctor Edward Pierce el próximo jueves. Espero que disfruten mucho esta actividad, que nos sigamos formando en otros temas que nos apasionan a todos y a todas los miembros de nuestra sociedad. Y bueno, los voy a dejar ahora con Cintia, que va a hacer la introducción formal del Dr. Ho. Muchas gracias por su presencia y espero que disfruten el seminario.
Buenos días. Bueno, voy a introducir al Dr. Ho. Hello, everybody. My name is Cintia Stempin. I am a researcher from CONICET at the University of Córdoba, Argentina. I have the pleasure to introduce our guest speaker to this immunometabolism webinar, Dr. Ping-Chih Ho. Dr. Ho got his bachelor's degree at the National Taiwan University and his PhD at the Department of Pharmacology, University of Minnesota. He performed his postdoctoral training at Susan Caich Labs in Yale University, where he started his studies on T-cell metabolism. In 2015, he was recruited as adjunct Ludwig scientist and tenure-track assistant professor in the Department of Oncology at the University of Lausanne, Switzerland. Then he was promoted to tenure associate professor and associate member at the Lausanne branch of the Ludwig Institute for Cancer Research, where he is directing his lab. His research is focused on understanding how tumor cells evade immune system through their metabolism and how metabolic machinery of immune cells can be reprogrammed to improve cancer immunotherapy. In addition, his work is also focused on delineating links between metabolic processes, signaling cascades, and epigenetic programming in the activation and differentiation of T cells and macrophages. His work has been published in the most prestigious journals such as Nature Immunology, Nature Medicine, Cell, among others. Despite being a young researcher, Dr. Ho has received several awards, including the Swiss Bridge and Fuller Cancer Research Institute Investigator, Melanoma Research Alliance, Young Investigator Awards, among others. Thank you very much, Dr. Ho, for joining us today.
Thank you. So maybe I can share my screen first. So first of all, I would like to thank you for Mariana and Luciana's invitation, and also the entire Immunometabolism Research Interest Group invitation to let me have the chance to present our research work to you. And also I would also like to thank Cynthia's very kind introduction to kind of go through my research career. So today I will talk to you about the research topics we did in the past few years, and then this would be a combination of two research topics. But basically, these two topics will mainly try to understand how metabolic hurdles happening in the tumor microenvironment can prevent effective anti-tumor immune response controlled by T cells, and how we can target this process to reprogram the metabolic fitness in T cells, which allowed us to elicit more robust and durable anti-tumor response for immunotherapy. So basically, I would say that if we want to achieve very strong anti-tumor response, we have to take care of two major actions for T cell reactions. The first one is that we have to bring T cell into the tumor microenvironment, and those tumor-specific T cell can also need to be activated properly in a tumor microenvironment. So basically, the infiltration activation should be taken care. However, this is not that simple when we talk about the tumor microenvironment, because in the tumor microenvironment, microenvironmental challenges listed here imposed by tumor cells that provide different layers restriction to hamper T cell anti-tumor response. For example, the first one, the metabolic restriction and hypoxia, they can shut down the T cell metabolic fitness and also create some hypoxic signature, which may be bad for the T cells. And I will cover that one later in my talk. And the second one is that actually the metabolic stress and also restriction happening in the tumor microenvironment can also prevent T cell infiltration. At the same time, this can also facilitate the expression
Dr.
of
Ho,
inhibitory.
excuse me.
Sorry.
I think,
yes.
okay.
We are still in the first slide.
I think you're not presenting the whole screen also, maybe?
Yeah, I present the whole screen.
Let's see.
Yes.
Do you see the whole screen?
No.
No.
So maybe let's redo it again.
Yes,
give me
please.
1 second.
Thank you.
Do you see it move now?
No, we are not seeing in full screen mode.
Okay, so maybe that's.
And we didn't see those slides, I think, this.
Okay, so give me 1 second.
Sure. No problem.
No problem.
It must be some setting issue. Okay. Okay, let's see. Can you see my screen?
We can see the screen, but not full screen.
How about now? It's full screen?
Not yet.
No.
No.
Okay, what happened? So maybe let me try to find out if this is any particular issue of setup. I need to check how to change it. Does anyone know how to change it?
Juan, maybe you know. I don't know. But don't worry, just we will try to, we can wait, don't worry.
Okay,
this is because we talk a lot before trying the presentation. We started to talk.
Okay, give me 1 second. I need
if
to know
not,
that.
The most important thing is to be sure that we can change this, that you can change the slides. If you can.
Yes, but I think I can change my slide in my end, but I just don't know why it doesn't change. Give me 1 second.
Do you want to send us the PowerPoint presentation? Maybe
yes,
Cynthia or Luciana can.
Yeah, sure.
Yes, I can do it now, but it will be quite big.
Ah, okay.
Let's take a
But
look.
we can wait, don't worry.
So I will send to you, Luciana. Just in case. Give me 1 second.
Sure, no, don't worry.
So Luciana, I will just send to you so that we
Okay.
can take a look. So troubleshoot.
That's strange. We didn't have any problems with other presentations.
I get the file and download it.
They are saying to me that maybe if you try to share the screen, let me see exactly what they are saying to me. If you try to share the screen to the one that it appears up left, you will see what you are seeing. I don't know if you have a possibility to see.
Yeah, but for me, it's fine for other presentations, so I don't know what happened here. So you see my screen again, right?
Yes. Let's see again.
See.
Let's try.
Okay, give me 1 second.
When you share there,
I do the
see
slideshow. Now it's full screen.
Well, see, we don't see it full screen. But let's see if you can move the slides. If we can see, there we go. At least we are seeing now that you're
Yes, so I don't know
changing the slides.
If I do this way, it's full screen.
No.
So, yeah, so something is
okay.
wrong.
In the meanwhile, I'm downloading the file that is taking.
So maybe Luciana, can you show the slide and then let's see whether this would not be a problem.
Yeah,
okay.
No problem. Just one minute and it will be completely downloaded.
Sorry about that.
No, don't worry.
Don't worry, don't worry.
Virtual issues. A few more seconds, please, and it will be ready.
Maybe I try again. I'll just modify some setup. So you see my slide again, right?
Yes, we can see your
slideshow.
Slide.
Is this one full screen?
No.
No.
That's weird.
That's weird. Really, it's weird.
Okay.
Luciana, have you been able to download the talk?
Yeah, but it is true that it was a little bit big, so I'm still.
Okay. Maybe we can
I'm open.
with.
I'm open.
It's okay. Okay. All right.
I'm about to open it.
It's fine. So it's introduction. I still can continue just while Luciana is opening the file. So
that's okay. Okay.
Okay.
There we go. I think everything will.
Okay, chat, let me know if you can see it full screen or not. If you need.
There we go. I think it's opening.
It's
perfect.
Okay.
Perfect.
Okay.
Great.
Sorry about that.
So let's start all over again. Perfect.
Okay. Sorry about that. So it must be some default setting issue. So anyway, so can you go to the next slide? So this is what I just mentioned, that actually, if we want to launch a proper anti-tumor response, improved infiltration and activation of tumor-specific T cells in tumors are critical. And this is the idea is very simple, because we want to bring soldiers to the battlefield and then also activate and trigger the action of those soldiers in a proper way. However, the microenvironmental challenge listed on the right imposed by the tumors actually present the major hurdles to block the infiltration and activation of tumor-specific T cells. The first one, the metabolic restriction and hypoxia, they are very common in most of solid tumors, and this will affect the T cell activation and also their action. And I will describe how this action can be done later in the first story I will share with everyone. And the second action is that actually the microenvironmental challenge can also impair the infiltration of T cells. And even those T cells have a chance to infiltrate into the tumor microenvironments, the metabolic reprogramming actions in tumor cells, they can also facilitate the expression of co-inhibitory receptors that can further shut down T cell actions. And the last point actually can be imposed by the tumor microenvironments is that the unknown regulation, which facilitates the accumulation of immune modulatory cells, including regulatory T cells and myeloid-derived suppressor cells and onto macrophage, as well as tolerogenic dendritic cells. So it has been suggested actually the microenvironmental regulation can play a very important role, which facilitates the aberrant accumulation of those immunomodulatory cells. So basically, I will also have the chance to cover the third point in my talk in the later part, and then try to let you know how we can use metabolic intervention to target both the point 1 and also point 3. Luciana, can you go to the next slide? Okay, so before I jump into the data we have, I want to let you know one of the major problems for anti-tumor immune response is that those tumor-specific T cells in the tumor microenvironment, a lot of them, they will enter to a very special differentiation state we call T cell exhaustion. And T cell exhaustion represents a major problem for the therapeutic benefits of immune checkpoint blockade treatment. As you can see here, when T cells become fully exhausted, they upregulate inhibitory receptors, for example, PD-1. At the same time, they also become non-responsive to immune checkpoint blockade treatment. This is mainly due to fixed epigenetic reprogramming that block the particular locus, which is important for effector function in those T cells. However, when T cells they are partially exhausted, even though they express PD-1, they still have the chance to respond to PD-1 blockade treatment. And if those cells they are effector T cells, they can also maintain their responsiveness to PD-1 blockade treatment. Can you go next? So this is what I say, the epigenetic landscape change is a very critical event to lock the T cell in the fully exhausted state. However, the underlying mechanism remain unclear how those tumor microenvironments over the chronic antigen exposure can trigger this epigenetic landscape change. Okay, go to next. So in order to understand whether the microenvironmental challenge imposed by the tumor microenvironment can be the driving force for this epigenetic landscape change, and also from the metabolic perspective, my postdoc decided to ask, so we know that actually the tumor microenvironment can be glucose deprived. They also have a lot of hypoxia region. So for any single given cell, if they experience those metabolic challenge, they have to change their metabolic preference. And mitochondria become very important organelle because this is an organelle to maintain our metabolic flexibility. Due to the TCA cycle, and cells will be able to choose whether they can use glucose or amino acid or fatty acid as a nutrient. So we decided to take a look whether the mitochondria in those T cells in the tumor microenvironment really change their property. So we isolate splenic CD8 T cell versus the tumor infiltrating CD8 T cell from tumor bearing host. As you can see here, based upon the electron microscopy analysis, the tumor infiltrating CD8 T cell actually contain a lot of mitochondria. And we can also quantify. You can see that the mitochondrial number per cell actually is much higher in the tumor infiltrating CD8 T cells. Next. However, when we isolate those T cells and perform qPCR to see whether they also increase their mitochondrial DNA, we didn't see the proportional increase. So as you know that mitochondria is inherited from a maternal part, so each mitochondria also contains their own version of mitochondrial DNA. So in this data, we see that actually the mitochondrial mass increased. However, a lot of them looks like they don't have mitochondrial DNA. That's why we don't see the increase of mitochondrial DNA copy number by the qPCR. And this is a typical sign of mitochondrial dysfunction. And this can be further confirmed when we use higher magnitude analysis for this EM. As you can see here, compared with splenic CD8 T cells, those tumor infiltrating CD8 T cells in the tumor bearing host, even though they have more mitochondria, but their mitochondrial morphology looks pretty weird. They don't have proper 3D structure. They don't have the chance to maintain very clear double membrane, which is quite important to maintain the mitochondrial proton gradient and mitochondrial membrane potential. Next. So with this information, we further ask whether those mitochondria displayed by tumor infiltrating CD8 T cells, they really have some issues to maintain their structure. Because when we perform EM, we only see the morphology and those information from one layer of section. So you probably will be biased based upon which layer you choose. So in order to avoid this bias, we decide to use the 3D EM. So we dissect every single layer of each cell and then superimpose all the image together. And in this case, we will be able to see the mitochondria in one single cell and see their connectivity and also have more detailed look of their cristae and also their structure. As you can see here, the tumor-infiltrating CD8 T cells, compared with the splenic CD8 T cells, those tumor-infiltrating CD8 T cells actually have more elongated mitochondria. Next. And actually, in each color is one mitochondria they have. And you can also see that actually in each mitochondria, their crystal valent, which is the indication of their folding and how the mitochondrial electron transport chain have the chance to interact with each other. This crystal valent actually also drops. So that suggests that actually those tumor-infiltrating CD8 T cells, they have more mitochondria. However, their mitochondria cannot fold properly. They don't have the chance to maintain the folding structure, which is important to maintain mitochondrial membrane potential and also for the assembly of electron transport chain to control the respiration in the cells. So when we analyze those data, we also realize that in the tumor-infiltrating CD8 T cells, we can probably distinguish them based on the morphology into two populations. The first population look like the left-hand side. They still have relatively normal crystal, as you can see here. And those populations, they also have a vacuole-like structure compared with the right-hand side. Those populations, they completely, display completely rubbish mitochondria. Their mitochondria doesn't have any good morphology. And you also see some rupture in the mitochondrial membrane. So when we realize this point and also see that the one with relatively healthier crystal contain the vacuole, we start to speculate probably those populations on the left, actually they have a chance to engage autophagy, which help them to maintain the mitochondria fitness. Next. So we take a look, or we next take a look whether we can see these two populations by using flow cytometry. So we use two different molecular probes, and actually we can also see two populations based upon the molecular probe, their property. The first probe we use is mitotract green. This is a mitochondria membrane potential independent molecular probe, so which can indicate whether you have more mitochondrial mass. And the other probe we use is mitotracker dip rate. It's the y-axis. And this is a mitochondrial membrane potential dependent dye. So once you have more mitochondria and you also increase the mitochondrial membrane potential, you will see that both mitotracker dip rate and mitotracker green increase their intensity. So based upon this co-staining, we can see two populations. The one labeled in green is the one I want to show you and let you pay attention. This one we call it NDRMG low. They contain lower mitochondrial membrane potential per mitochondrial mass. And this is the population usually you will see in aging T cell, aging neuron cells, and aging muscle cells, because they start to accumulate more mitochondria, but majority of the mitochondria they have actually have less mitochondrial membrane potential. And this low population actually increased in tumor-infiltrating CD8 T cell compared with CD8 T cell in spleen or draining lymph node from the same tumor-bearing host. Next. And we can also see this population increase in patient samples. So we also analyze human melanoma patient samples, and each line in this data indicates a pair sample. So the CD8 T cells in PBMC versus CD8 T cell in tumor. As you can see here, majority of patients we analyze, they always contain higher mitotracker dip rate versus mitotracker green low population in the tumor microenvironment, which suggests that actually in the tumor microenvironment, most of the TLs, CD8 T cells you observe over there, they will start to accumulate certain damaged mitochondrial phenotype. Next. So I told you that actually we see that there are some vacuole structure in those T cells with relatively healthier mitochondria, and we speculate this is due to the autophagy defect. So how autophagy can control mitochondrial fitness? And this is pretty simple because autophagy can control a very special mitochondria dynamic process called mitophagy. And this is a process which is quite important when we start to use mitochondrial activity in a maximal level. So for any single given cell, if they are under nutrient restriction, for example, in a glucose deprived condition, or in a condition they have to engage stronger fatty acid oxidation, they need to use oxidative metabolism to fit their energy demand. They will fuse their small mitochondria and form a big mitochondria. Next. And in this case, the reactive oxygen species will accumulate. and which will damage the mitochondria, and you will start to see some mitochondria will have the oxidation on a mitochondrial membrane lipid or in a protein. And normally, actually, we can remove the damaged parts through the mitophagy. Next. So this is the process. We use the cells will use the autophagy machinery to engulf the damaged parts and then recycle the ingredient, and they will keep the functional part for further usage. However, when cells they are aged, including neuron and muscle cells, they will have difficulty to engage this mitophagy machinery. And as a result of defective mitophagy, next, those cells will start to accumulate damaged mitochondria. And then once the damage is too severe, those mitochondria lose the mitochondrial membrane potential. So that's why you will start to see a cell that start to contain a lot of mitochondria. But those mitochondria that accumulate, they start to show no mitochondrial membrane potential or reduced mitochondrial DNA. And this will change the cellular behavior. So with this information, we start to speculate those MDR-MG low population we observe in the tumor microenvironment, indeed, they have the problem to engage mitophagy. And since this paper already published at the end of last year, so I will not go through the detail. Actually, we performed several kind of cellular biology experiment to confirm that those T cells, if they lose their mitophagy machinery, they will accumulate more MDR-MG low population. And the accumulation of the low population actually indeed is controlled by the defective mitophagy machinery. So the question we decide to ask next is that once T cells in the tumor microenvironment, they accumulate damaged mitochondria, can this be a problem? Can this drive those T cells become so-called terminal exhausted T cells? So in order to examine whether those cells become terminal exhausted T cells, we have to analyze three categories. Because examining their PD-1 is not sufficient. PD-1 can be an activation marker, and PD-1 high or low sometimes is pretty, I would say it's not a clear cut to indicate the T cell functionality. So when T cells become terminal exhausted T cells, they will lose their proliferative capacity when you restimulate those cells. They will sustain their inhibitory receptors, including PD-1 and TIM3 expression. At the same time, they will also sustain low expression of cytokine because those cytokine locus has been permanently closed by epigenetic mark. That's why once they receive the following activation, the signaling cascade is not sufficient. Even you use PD-1 to restore the signaling cascade, they still cannot produce cytokine. So in order to extend these three categories, we isolate splenic OT1 from tumor-bearing mice and also OT1 TILs based upon their mitochondria phenotype. So we have the red one is the mitochondrial activity sufficient population. The blue one is mitochondrial activity deficient population, which accumulate a lot of damaged mitochondria. And then we transfer that into naive mice with the same amount into individual one. And one day later, we challenge those mice with listeria ova because those OT1 will recognize ova, so we can examine whether the T cell will start to re-expand, whether they can keep their proliferative capacity. And we can also know that whether the T cells they expand will sustain the inhibitory receptor expression because ovabulin listeria infection usually will not trigger sustained PD-1 expression because T cell will have a chance to clean the pathogen. So they will not engage sustained expression of PD-1. And normally this rechallenge will make the T cells start to produce cytokine. So based upon this experiment, we show that we found that next. The low population, actually, they proliferate much lower compared with splenic OT1 or OT1 TILs with sufficient mitochondrial activity labeled in red. Next. And this low population actually sustains the expression of PD-1 and LAG3. Next. In contrast, they don't produce interferon gamma as efficient as splenic OT1 or OT1 TILs with sufficient mitochondrial activity. So with this information, we can basically conclude that when a T cell in a tumor microenvironment accumulate damaged mitochondria, they will start to display terminal exhausted T cells phenotype, including reduced proliferative capacity, sustained inhibitory receptor expression, and sustain the declined production of effector cytokines. Next. And as I told you, we also know that based upon chronic viral infection and also other human tumor models, those terminal exhausted T cells will display different epigenetic landscape compared with functional T cells. The epigenetic landscape including the chromatin occupancy and also the DNA methylation pattern. So in order to see whether we also observe those changes people already report before, they can control the T cell, they associate with T cell exhaustion. So we isolate those OT1 TLs with different mitochondrial activity, and then perform whole genome bisulfite sequencing. As you can see here, this is the illustration of CpG island methylation pattern. The one labeled in red, they have more methylated CpG island in each particular locus, okay? So you can see here, the low population compared with high population, they display distinct methylation pattern. Next. At the same time, if we perform a tag-seq, we can also see robust difference in the chromatin architecture because the low population start to close certain locus and open particular locus. And the locus they close actually you can see that the TCF1, which is important for the stem-like property in T cells, become closed in the low population compared with the mitochondrial activity sufficient T cells. And the regulator of TCF1 expression, including LEF1, is also lower. The chromatin occupancy is also lower. So with this information, we confirm that once T cells in a tumor microenvironment accumulate damaged mitochondria, they would display, in terms of functional features, look like terminal exhausted T cells. They also display DNA methylation pattern, which resemble terminal exhausted T cells, as well as the chromatin architecture will also change into a way look like terminal exhausted T cells. Next. So with this information, we next try to understand what kind of factors in the tumor microenvironment can be the driving force to facilitate the accumulation of damaged mitochondria. And our first guess is that actually the T cell receptor signaling should be the driving force, because in the chronic viral infection or in the tumor setting, those two settings, actually we can see T cell exhaustion. And people believe that this is due to the persistent antigen exposure. So with this information, we decided to take a look whether this is the case in the mitochondrial regulation. So what we did was very simple. We engrafted melanoma cells into two different flanks of the same mouse, and they are the same melanoma cells, but those melanoma cells express different antigen. So on the left-hand side, this is the melanoma cells express ovalbumin. On the right-hand side is the melanoma cell express glycoprotein 33 of LCMV. So we co-engraft the same amount of cells into two different flanks and wait for 13 days. And then we co-transfer OT1 plus P14 into those tumor-bearing mice. And OT1 will recognize ovalbumin, overexpressing melanoma cells. P14 will recognize GP33 peptide, presented by the MC class 1. And we wait for 5 days and see what happened for those cells in terms of their mitochondrial activity. Next. As you can see here on the left-hand side, in the ovar tumor, you will see OT1 cells display mitochondria, MDR versus MG low population, as indicates in the flow plot. But P14, you don't see those population in the ovar tumor. In contrast, in the GP33 tumor, you will see more P14 accumulate damaged mitochondria compared with OT1. And the right-hand side is the quantification result. Each line indicates pair sample from one mouse. And no matter which mouse we analyze, we always see the same trend. In the ovar tumor, you will see OT1 accumulate damaged mitochondria. In GP33 tumor, you see P14 accumulate damaged mitochondria, which indicates when the T cell infiltrate into the tumor microenvironment, if they can recognize the tumor antigen, they will have higher possibility to engage certain problem, which leads to the accumulation of damaged mitochondria in the cytoplasm. Next. And by using, can you go back? By using other approach, we also found that actually affinity is also quite important. The T cells in the tumor microenvironment, if they have a higher affinity to the tumor antigen, they also accumulate damaged mitochondria way faster and way stronger than the one with weaker affinity. So based upon those information, we conclude two things. The tumor microenvironment and the T cell receptor signal are needed to drive the damaged mitochondria formation in tumor infiltrating CD8 T cells. And the T cells you have, which recognize tumor antigen with a higher affinity, they tend to accumulate more damaged mitochondria. So this actually creates a very important issue, which suggests that your best soldiers will be the most vulnerable population. They cannot maintain their mitochondrial fitness. Next, and this is also true based upon the single-cell RNA sequencing in human samples, because people already know that in single-cell RNA sequencing samples, the one which recognizes the tumor antigen with the higher affinity based upon TCR clonality, they actually display more terminal exhausted state. But no one really knows that's what's the reason behind that. And based upon this study we presented end of last year, we demonstrate that actually the T cells in the tumor microenvironment, in the beginning, they probably have the chance to maintain their mitochondrial fitness. In this stage, they will have a good metabolic fitness because their mitochondria can control their flexibility to use different nutrients. At the same time, they will also have better ability to proliferate and then to express cytokine. However, in the tumor microenvironment, next, they will encounter two different signals. One is the coordination of hypoxia and chronic TCR stimulation, which will block autophagy and mitophagy machinery. Next. The second signal is glucose deprivation. By combination of glucose deprivation and hypoxia, those cells will start to use error-prone metabolic process, which facilitates the mitochondrial damage. So you can consider that the first signal block your mitophagy, but the hypoxia plus your signal 2 will create damage. So overall, you will see those cells start to have a lot of damage they cannot remove, and they will start to enter to the next phase. Next, they will have a lot of damage mitochondrial accumulation in the cytoplasm, which facilitate epigenetic reprogramming and sustain the co-inhibitor receptor expression in those cells. And eventually, those T cells become terminal exhausted T cells. They will not be able to respond to PD-1 blockade treatment. They also start to engage epigenetic landscape alteration. Next. So with this information, we decided to take a look. Can we find out any treatments that can block this commitment? So if we can find out this treatment to block this commitment, to prevent this accumulation of damaged mitochondria, we might be able to preserve T cell responsiveness to checkpoint blockade treatment, and also preserve their functionality, and also preserve the population of progenitor exhausted T cells. So with the in vitro system we set up, we can induce the low population formation by giving T cells cancer cell conditional medium, because this is glucose deprived, and also sustain T cell receptor signaling plus hypoxia. However, this low population accumulation can be prevented if we add nicotinamide riboside into the culture. And nicotinamide riboside actually, it's a NAD supplement. It has been shown to promote autophagy and mitophagy due to the 33 action. So with this information, we actually suggest that this food supplement, nicotinamide riboside, can be a good candidate to prevent the commitment of terminal exhaustion in tumor-infiltrating CD8 T cell. Next. So we also demonstrate that actually we can package this nicotinamide riboside into liposome, and then by intratumor injection, we will be able to slow down the tumor growth and also improve the PD-1 responsiveness to those tumor-infiltrating CD8 T cells, as you can see here. Okay, so this data suggests that actually the metabolic restriction happening in the tumor microenvironment will create a lot of problem for those CD8 T cells in the tumor microenvironment. And as a result of this metabolic stress, CD8 T cells will not be able to maintain their mitochondrial fitness. And this can further lead to epigenetic landscape change and then the commitment of terminal exhausted T cells. If we can take care of these parts, then we'll be able to sustain T cell functionality. Next. So this study, the first study I want to show you suggests that actually the mitochondria become a very important powerhouse, not only provided energy to the T cells, actually they also provide a very important retrograde regulation to ensure those T cells can engage particular epigenetic and transcriptome they should display based upon their metabolic status. Okay, next. And the main reason is that probably the mitochondria it's important to control the production of different metabolites, including acetyl-CoA and also lactates, SAN, and also alpha-ketoglutarate succinate fumarates. They can control the epigenetically modifying enzyme expression. And those metabolites I mentioned, like acetyl-CoA or SAN, they can control histone acetylation and histone methylation and DNA methylation. So we think that the functional mitochondria in T cells will ensure that those cells will be able to produce the critical regulators they need to control epigenetics. So we still don't know when the T cell they start to accumulate damaged mitochondria, which regulator will start to have trouble, and how come this will drive the gene locus specific regulation. So we are still in the process to understand the mitochondria and nucleus crosstalk in CD8 T cell exhaustion condition. Next. So this will be the first example I want to show you that actually what happened for CD8 T cell. But if you remember what I told you in the beginning, immunomodulatory cells, they can accumulate in a tumor microenvironment and do their job to suppress immune response. So it becomes a very interesting idea. How can CD8 T cells or those T cells, those immune cells that try to kill cancer cells, they cannot maintain their metabolic fitness. But those immunomodulatory cells, those bad guys, they can do a good job in the same environment. So there must be something happening in those bad guys, which make them become super unique in the same environment and behave normally in the same environment. So I would like to switch the gear toward the bad guys. One of the major bad guys in the tumor microenvironment is regulatory T cells. As illustrated here, regulatory T cells not only the bad guy in the tumor microenvironment, actually it can play a very important role to control tissue homeostasis and immunohomeostasis in any single host, including mouse and human, because they can control the inflammatory process and avoid overt inflammation, which will destruct the tissue and also create certain autoimmunity problem. So those cells actually, they can also do good thing in a normal condition. However, next, in the tumor microenvironment, regulatory T cell will be aberrantly accumulate next. And this is known in multiple tumor types. And in those tumor types, if you see a lot of regulatory T cells, usually you can expect the microenvironment will be more immunosuppressive because regulatory T cell will just basically suppress all the immune cells action, including dendritic cells, macrophage, and T cells. At the same time, those tumors with higher Treg infiltration, they will also have poor prognosis and poor responsiveness to checkpoint blockade treatment. So with this information in hand, people want to develop treatment that allow them to target regulatory T cells in order to unleash host anti-tumor response. As you might be able to expect, regulatory T cell control immune homeostasis and tissue homeostasis. If you want to have any treatment target regulatory T cells, you would disrupt those homeostasis. And that's very easy to see autoimmunity in patients receive this kind of treatment. So this becomes a problem. This also limited the therapeutic window of Treg targeting approach for cancer treatment. Next. So that's why we want to find treatments that can selectively demolish intratumor Treg while keeping Treg in peripheral tissue, in lymph node, intact and functional. Next. So when we start to work on this idea, my student just proposed that probably we can find out the particular metabolic activity used by intratumor Treg. And we think this is so-called metabolic adaptation. The reason we have this kind of hypothesis is very simple. If I'm a Treg, you can find me in different places. For example, you can find me in Switzerland, you can find me in the States, you can find me probably in Argentina, but I'm still the same person. But in order to survive, in order to do my activity, I have to eat, but I will not be able to get the same food in every single place. I have to change my preference of diet. So as a Treg in different places, they probably also change their nutrient preference in order to survive. And they probably change their behavior based on what they eat. Okay, so this is the process we speculate. The metabolic adaptation can change the Treg behavior in different places. Next. If this is the case, we probably can target the metabolic preference only used by intratumor Treg. And this will reduce the therapeutic side effect because Treg in brain, Treg in spleen, Treg in muscle, they don't use the same metabolic process. Then we will be able to selectively demolish intratumor Treg. Next. Next. So my PhD student decided to take a look and we decided to learn from human patient samples. So we reanalyzed the data done by Sasha Rudensky In this paper, they isolate Treg from breast cancer patients from both tumor and also PBMC, and they focus on chemokine receptors. And what we did was to reanalyze this sample and then focusing on the metabolic enzyme expression between tumor-infiltrating Tregs versus Treg in PBMC. As you can see here, we see robust increase of genes controlling lipid metabolism in intratumor Treg. As you can see here, for example, lipid metabolic process respond to lipid binding, and lipid biosynthesis process. Those enzymes, those genes will be upregulated in intratumor Treg in this breast cancer patient cohort. Next. Among them, we also see that only one lipid translocase will be selectively increased by intratumor Treg, which is CD36, is responsible for medium and long-chain fatty acid uptake, and also for oxidized LDL uptake. Next. So with this information, we decided to take a look. We have to always bear in mind, RNA sequencing is always RNA sequencing, especially when we talk about metabolic regulation. RNA to protein to the metabolic activity, there's huge gaps in between. So we have to take a look whether we can really see those intratumor Treg increase the expression of CD36 in terms of protein before we move into the next direction. So we engraft cancer cells into wild-type B6 and then isolate Treg from draining lymph node, non-draining lymph node, spleen, thymus, skin, and tumor, as you can see here. And you can appreciate that based upon effects analysis, we indeed see more CD36 expression in intratumor Treg compared with Treg from other places. And this is also holding true in human. Next. When we perform the FACT staining in melanoma patients, you can also see that on the left-hand side is representative flow plot. Those tumor-infiltrating regulatory T cells express higher level of CD36 on the surface, compared with the Treg from PBMC. And the right-hand side, each line is a pair of samples. Most of the Tregs from tumor-infiltrating lymphocyte, they express higher level of CD36 compared with the Treg in PBMC. So with this information, we start to speculate CD36, which is responsible for the lipid uptake, may control the Tregs to get lipid they need for their tissue retention and for their tumor retention and also for their survival in the tumor microenvironment. Next. So in order to examine this possibility, we generate Treg-specific CD36 knockout mice by crossing FOXP3 YFP3 with CD36 flux mice. And FOXP3 is the key transcriptional factor to control regulatory T cell development. So once they become Tregs, they also express Cre recombinants. In this case, they can get rid of CD36. They can knock out CD36 locus. So we also use FOXP3-YP3 as a control. When we engraft cancer cells into those mice, we first check whether the tumor-infiltrating Tregs need CD36 to maintain the lipid uptake and lipid content. As you can see here in the bottom, on the left-hand side, this is the lipid content, lipid uptake, sorry. So those intratumor Treg, which don't express CD36, they will display lower lipid uptake ability compared with the wild-type Treg in the tumor microenvironment. And on the right-hand side is this lipid content. We also see that the lipid content reduced in those intratumor Treg which don't express CD36. Next. Next, and most strikingly, by knocking out one single gene in one single immune subset, we already reduced the tumor burden. So it suggests that actually Treg need to express CD36 in order to support tumor progression. Next. So if you remember what we postulated in the beginning, can we find any method to selectively target intratumor Treg, but keep Treg in other place intact? So with this information, we see a reduction of lipid uptake and lipid content in intratumor Treg when they fail to express CD36. We next ask whether we can see the abundance change. So as you can see on the top side, the representative FACT plot. In spleen and draining lymph node between wild type and Treg specific CD36 knockout, we don't see any difference in terms of Treg abundance between those between wild type and knockout in those tissues, based upon the FOXP3-YP3 staining. However, in the tumor microenvironment, you can start to see a 40% 0.7 to 29.3, this difference. And if we quantify all the difference, you can see in the bottom side, those Treg specific CD36 knockout mice, they will have 40% reduction of Treg abundance in tumor microenvironment. However, their Treg abundance in spleen and draining lymph node, they are very similar to the wild type mice. Okay, next. Okay, here. So we saw that actually by knocking out CD36, you can reduce 40% of Treg in the tumor microenvironment, but the Treg in other places, they are intact. But we have to be careful. You see those Treg, they still have the same abundance in spleen and draining lymph node. Doesn't mean those Treg, they are function. So they probably lose their particular suppressive activity, even they can show up in peripheral tissue. So we next try to evaluate whether the CD36 expression in Treg is needed for them to suppress inflammatory response in peripheral tissue, which is not tumor. So we use the T-cell transfer colitis system to analyze this possibility. So we can transfer naive CD4 T-cell into REG1 knockout mice. And due to the lymphopenia condition, the T-cell will start to expand, and you will see a lot of immune infiltrate into colon and small intestine in the bottom part, so you can see here. And this will change the entire morphology of colon and small intestine, and then those mice will start to have diarrhea and then reduce their body weight, as you can see on the top. And this entire IBD can be prevented if we co-transfer naive CD4 T cells with Treg. So no matter we transfer wild type Treg or CD36 knockout Treg, both of them have the same efficiency to prevent gut inflammation and also prevent the body weight loss. So it suggests that actually in the peripheral tissue, when the Treg, they try to control inflammation, they don't need CD36. It looks like they only need CD36 in the tumor microenvironment. Next. So with this information, we show that actually intratumor Treg need CD36 to maintain their accumulation. They also need CD36 to maintain the suppressive activity in tumor, but not in the peripheral tissue. So our next question is that why Treg in tumor, they need CD36 to maintain this, to maintain their abundance. So we tried the migration, proliferation, we didn't see any difference. So we start to speculate, maybe those T cells, those Tregs in tumor, they need CD36 because this lipid metabolism can support their survival. And this indeed is the case because when we perform RNA sequencing, we isolate those Treg from wild type tumor infiltrating Tregs versus CD36 knockout intratumor Treg. We can see that those CD30 deficient intratumor Treg upregulates genes controlling apoptosis. Next, and this can be further confirmed by staining cleave caspase-3 and annexin-5. Those intratumor Treg which don't express CD36 start to upregulate active caspase-3 and also annexin-5 staining, which suggests that without CD36, we already see 40% reduction of intratumor Treg abundance, but the remaining one, they are dying. Okay, next. And the main reason they die because they cannot maintain their mitochondrial activity. I will go through how we find out this process with you. So we also isolate those Tregs from wild type versus Treg specific CD36 knockout mice from spleen and tumor. As you can see on the left-hand side, those splenic Tregs between wild type and knockout, they are very similar in terms of mitochondrial number and also crystal number. But in tumor, you will see a totally different picture. Those CD36 deficient intratumor Treg, they have less mitochondrial content, and their mitochondria also have less crystal structure, which is important to maintain mitochondrial membrane potential. So it suggests that CD36 expression in intratumor Treg is quite important to maintain their mitochondrial fitness. They can support the Tregs to engage stronger mitochondrial activity, maintain mitochondrial biogenesis probably. Okay, next. So you probably will ask me, what happened? Why mitochondria? So if you remember what I told you in the first part of my talk, mitochondria is a very important organelle to maintain metabolic flexibility of every single cell. And in the tumor microenvironment, because the nutrient deprivation and also hypoxia and also lactic acid enrichment, so mitochondria become very important. If you don't have mitochondria, you probably cannot deal with those stress. So in order to examine whether this is the case, we set up the artificial in vitro system So we give those Treg we generate from wild type or CD36 knockout mice with different insult. As you can see here, we first give them RPMI culture medium, just a normal culture medium. We don't see any difference after 24 hours. The cell abundance is very similar. However, if we give them RPMI plus hypoxia, you can start to see CD36 knockout Tregs reduce their viability after 24 hours. The other condition we use is that we culture those Tregs in the presence of cancer cell conditional medium, which is lactic acid enriched. So we also see the difference. So with this information, we start to speculate. In a tumor microenvironment, there's a common factor between hypoxia and cancer cell conditional medium can kill Treg if they don't express CD36. And the common factor between hypoxia condition and cancer cell conditional medium, in this case, is lactic acid, because hypoxia can drive lactic acid enriched and production in the cell culture. Next. So we test whether lactic acid is the case. So we culture the Tregs, wild type and knockout Treg, and CD36 knockout Treg in RPMI with escalating dose of lactic acid. As you can see here, once we increase lactic acid dose, we start to see CD36 deficient Treg. They fail to maintain their survival in a higher sensitivity to lactic acid. Okay, next. And you may speculate what happens, why lactic acid is important, how lactic acid can, the lactic acid induced toxicity can be controlled by mitochondria. So I would like to bring you back to the biochemistry textbook. So if we culture every single cell in a lactic acid enriched condition for a long time, cell will die. And this is pretty simple because, for example, you culture cells, cancer cell or macrophage in the medium for 24 hours, you don't change the medium will become yellow. And if you continuously ignore it, the cell will start to die. The reason is that those yellow media indicates you have a lot of lactic acid in the media. And this lactic acid actually will flux in to the cell and it will acidify all the protein component and also cytoplasm. However, Treg can deal with this because they can convert lactic acid into pyruvate. As a result of this action, they will consume their NAD and generate tons of NADH. Okay, and normally this is not a problem for any single cell, and Treg can do that. But if they want to do this for a long time, for sustained durability, they will encounter a problem. They run out of their NAD. Next. So in order to sustain this entire metabolic process, they need to use their electron transport chain complex 1, so-called NADH dehydrogenase, to regenerate NAD. Next. And this will support this cycle again and again. And Treg will be able to support their survival in the presence of lactate for a long time. So we found that after CD36 knockout mice, they have less mitochondria. So we start to speculate they cannot maintain this cycle. Next. Indeed, that's the case. The CD36 knockout Treg, they have higher NADH versus NAD ratio, which suggests that they cannot regenerate NAD. And that's why they would die eventually if lactic acid is there too long. Next. So to test this idea, we set up this culture I showed you before. Again, if we give the cells with cancer cell conditional medium, you can see CD36 knockout Treg, they cannot maintain their viability. However, this reduction can be prevented if we give Treg nicotinamide riboside. As I told you before, this is a precursor of NAD. So by giving them nicotinamide riboside, we artificially resupply NAD, even they cannot generate that. Through the NADH dehydrogenase activity, we can artificially give them some extra components for the NAD production. In this case, they start to survive. So it suggests that the CD36 deficient intratumor Treg, they cannot maintain their survival in a lactic acid enriched condition due to the imbalance of NAD-NADH ratio. Next. Next. So for the sake of time, I will not go through all the detail because this paper also published last year. In this study, we actually find that in those intratumor Treg, they will upregulate CD36, which help them to get fatty acid from outside. But they don't really use fatty acid as a main ATP source. Instead of that, they use fatty acid to activate PPAR pathway, which is important to control mitochondrial biosynthesis and also mitochondrial activity. Next. As a result of this action, those Treg will be able to deal with lactic acid enriched conditions, such as a tumor microenvironment. Next. But if you block CD36, those Treg would not be able to maintain their PPAR pathway. They will not be able to maintain their mitochondrial biogenesis. They will fail to deal with lactic acid because they will not be able to recreate proper NAD/NADH balance they need to deal with lactic acid enriched condition. Next. So this would lead to cell death. Next. So with this information, we also start to speculate. We find out the metabolic adaptation intratumor Treg need, but other Treg they don't need. Can we target this process to launch stronger anti-tumor response? So we find out an anti-CD36 antibody, which can bind the lipid binding site of CD36 to block the lipid uptake. By injecting this antibody, we can already reduce the tumor burden. Next. And also reduce intratumor Treg abundance, but sustain the Treg abundance in spleen and draining lymph node. Next. And those intratumor Treg we can still identify in the tumor microenvironment. They will upregulate active caspase-3 staining upon CD36 antibody treatment. Next. Most importantly, when we treat melanoma bearing mice with anti-CD36 antibody, we can already slow down the tumor progression, as you can see, the red versus the black one. But if we combine anti-CD36 antibody with anti-PD-1, we basically stabilize the disease in this inducible BRAF-PTEN melanoma system. Next. So based upon this data, we identify a very unique metabolic process which support immunomodulatory cells, in our case, is regulatory T cells, to survive and stay in the tumor microenvironment. And we also demonstrate our hypothesis is correct. If we identify this metabolic adaptation, we will be able to identify unique therapeutic approach that allowed us to selectively target intratumor Tregs while keeping Treg in other peripheral tissues intact and function. And this will become a very important foundation for us to develop anti-CD36 based immunotherapy. Okay, next. So in the end, I would like to thank all the people in my lab, especially my the first story was done by my postdoc, and the second story was done by my PhD student, and also all the collaborators. They contribute into the different analysis and also human sample in this entire two study. And most importantly, the funding source. Without their support, it's not possible to conduct those animal experiments. And I'll be happy to answer your question. I also apologize for the beginning of the presentation issue. Thank you.
Okay, thank you. Thank you very much, Dr. Ho, for your incredible talk. I really think that the way that you tied together immunology, metabolism, and cancer biology was really amazing. And I think it was very useful for a broad audience. So I'm sure we will have a lot of questions. So that's why we will now jump into the questions and answer session. Please remember that all participants can make questions by posting them in the YouTube chat box. So let's see. Okay, so the first question that we have is whether are there 3 intratumoral cells better adapted to the tumor microenvironments than effector cells? What happens to the expression of CD66 in CD8 lymphocytes?
Okay, so maybe I can ask, this is kind of two questions in one. So first of all, Treg can adapt much better than CD8 or other CD4 T cells for lactic acid. This is a mystery, I would say, but it has been shown recently in vitro paper in cell metabolism. They show that actually FOXP3 expression in Treg will change the metabolic program of Tregs. So they can help them to survive much better in lactic acid enriched condition. So without FOXP3 expression, other T cells cannot do that. So I would say that's the reason Treg can adapt much better than CD8 and other CD4 T cells, because they have a very special transcriptional program support their metabolic need. Okay. And for CD8, the CD36 expression in CD8 T cells, this is a very interesting question. So I collaborate with my postdoc mentor, and this paper got published this Monday. And there's another paper in cell metabolism also show that CD8 T cell in the tumor microenvironment also upregulate CD36. But the expression of CD36 in those CD8 T cells, they don't do good things for CD8. They will promote ferroptosis. They also promote T cell exhaustion. So in those two studies, both of them demonstrate targeting CD36 can restore CD8 T cell functionality and anti-tumor response. If you combine their finding with our finding, we can suggest that actually targeting CD36 can kill two birds in one stone. You destroy Treg, you restore CD8 T cell responsiveness. But the mystery will become why CD8 T cell cannot behave like Tregs. I think probably get back to the first answer, FOXP3.
Great. We have another question from the first part of your talk. Sustained TCR activation would somehow induce a defect in mitophagy?
Okay, so we don't really know the answer, but our first guess is that the TCR stimulation will block mitophagy due to the engagement of mTOR activity, because TCR will stimulate mTOR, and higher mTOR activity basically abolish autophagy and mitophagy. And this hypothesis will be partially demonstrated by a new paper published in Immunity, I think probably in 2 weeks. Because in chronic viral infection, I know that people also demonstrate this is true. And this is due to the dysregulated mTOR activity, which drive the mitochondrial phenotype change in T cells.
Okay, before leaving this question, I would like to ask you something related to that. So, because do you think that this failure in the autophagic process is specific for mitophagy, or maybe the clearance of other damaged organelles or vesicles can also be affected? I was, for example, thinking about lipid droplets and lipophagy. Is it also altered in these TILs?
I'm not really sure, but I assume it should be, because we use mitophagy reporter, the one I didn't show. We also use autophagy reporter. Both of them will be abolished. Both autophagy and mitophagy will be abolished in those CD8 T cell in the tumor microenvironment. And so this suggests that the fundamental machinery, the autophagy machinery, is controlled. It's kind of tightly controlled in those CD8 T cells. And if they don't have the chance to engage that, they will have the issue. And I assume this will also control the way they deal with the lipid droplets, the way they deal with other organelle homeostasis.
Okay. So next question is, do you have any hypothesis regarding the expression of LEF1 and how it is related to dysfunctional mitochondria?
That's an excellent question. So I didn't really touch that, because LEF1 and TCF1 expression define whether the cell will become progenitor exhausted T cells. So our data suggests that if they lose their mitochondrial fitness, they will not be able to sustain their progenitor phenotype. And this is true in an LCMV. You will see the paper, as I say, in 2 weeks. And this is also true in HCV and HBV, because I know that people in that field, they already see the same thing as we report. But the mechanism which link the mitochondria to the progenitor phenotype is not clear. We have few hypotheses, for example, DNA damage response or BLIMP1 expression, but we don't know the entire picture yet. But we are in that direction, try to understand how this link can be done.
Okay. Do you expect, or have you been able to evaluate if mitochondrial dysfunction is reversed in TILs from patients treated with immune checkpoints inhibitors such as anti-PD-1 or PD-1?
So I don't think,
yes.
I would say that mitochondrial dysfunction can be reversed. It's very easy to reverse it. Okay, so for example, in our case, we can see the MDR-MG low population in a tumor. If you isolate those population and you calculate in vitro, after 24 hours, they become normal in terms of mitochondrial activity, but their functionality will not be normal. So it's also similar to what we did. We even isolate those population and then transfer into naive mice. Their mitochondria become normal, but their functionality doesn't come back. So it suggests that when the damage accumulates to a certain level, once the cell decide to engage epigenetic landscape change, it will be too late. And in this case, PD-1 blockade doesn't help. And people already know that in LCMV, in tumor.
Okay, our next question, well, it is related to the first one. Are all fields similarly susceptible to levels of tumor lactic acid and Treg, or only tumor Treg? Other fields do not use CD36 for the metabolic balance.
Are all the teams similarly accessible? I don't think so, but people can only do that in vitro. I know that, for example, the study I described, they published in Cell Metabolism with the in vitro data. They show that actually Treg can survive much better and proliferate in the presence of lactic acid. But CDA, they don't. Once you calculate in lactic acid enriched condition, their proliferation just decrease, and NK cell cannot. So for some reason, Treg can deal with this lactic acid. They will not be, their glycolysis will not be abolished by lactic acid. In our case, we really think CD36 is the key, because CD36 will help them to convert lactic acid to something else. If they don't have the CD36 signaling cascade, they will not be able to convert lactic acid into pyruvate, and they will have the same susceptibility to lactic acid induced toxicity and proliferation arrest like CD8 T cell, like NK cell.
Okay. So I would like to add last question, because when you treated the mice with, I guess, an error, so my question is whether this treatment can also affect the cancer cells themselves, because as far as I know, it is used sometimes to fuel glycolysis. And it was, I don't know, I was thinking whether this can also affect directly the tumor cells, not only.
So this is the question actually our reviewer asked us.
It was me.
Yeah. So we got this question before the pandemic, and then we started to do the experiment, and then pandemic happened, so we kind of kill every mice. But so that's why we don't really answer this. We didn't really answer this question sufficiently enough. So we use another way, kind of due to the time limitation. So we culture the cancer cells and give them nicotinamide riboside, and we know that they will see certain change, but nicotinamide riboside will not directly kill cancer cells. Okay, and we also know that the nicotinamide riboside induced anti-tumor response, as we see in the figure I show you, is CD8 dependent, because once you get rid of CD8 T cell, you will not see any beneficial effect. So it more or less suggests that nicotinamide riboside might directly affect CD8 T cell. It should be a direct effect rather than the indirect effect coming from the cancer cells. But we cannot rule out other players. It's quite difficult.
Thank you very much. I don't know whether.
Can I answer the last one?
Sure.
Have you had the chance to explore mitophagy? The ubiquitin ligase pathway or not, or the other one in your TILs?
You mean which one is defective, right?
Yeah, right.
We don't know. First of all, the mitophagy is a very complicated machinery. Right now the most dominant pathway, including Parkin, Nix, and there are a few more actually. So there are multiple pathways can control mitophagy. We are not really sure which one we should focus, but at least in our study, we also demonstrate that if those T cells, they don't have Parkin, they will accumulate damaged mitochondria way faster than the wild type. So it suggests that mitophagy machinery is needed, but I don't really think Parkin is the major one controlled by, it should be used by T cells. Some other mitophagy machinery should be there, but we don't really know which one.
Yeah, it is complicated.
It's very complicated.
In T cells especially.
Exactly.
So we have one further,
there's also one that called FUN-DC, because FUN-DC is also very highly expressed by CD8 T cells.
Yes.
Yeah, we have one further last minute question for you. It's from Antonio Cabrera Silva, Javier Cabrera Silva. He's asking whether, well, he said that maybe he missed the information, but whether by depleting CD36 into REX, if you also get a reduction of exhaustive markers on the factor CD8.
We don't see reduction of exhaustive markers. You know, it depends on how you define it. We don't see reduction of PD1. We don't see reduction of LAG3 in CD8 TILs, but we see better cytokine production. Okay, so that's why, you know, in the tumor immunology field, people need to be careful. When the T cell express PD1, sometimes they are exhausted, sometimes they are not. They just, you know, like a feedback mechanism, they have to upregulate PD1. So test their functionality and their proliferative capacity is a better way to confirm whether they are exhausted or not. Or the other way you can do is to check their tox expression. Maybe that's a relatively better way than just measuring PD1. But the short answer is that targeting CD36 in Treg, we do see better polyfunctionality in CD8 TILs. And that's why we think PD1 blockade can work much better when we introduce anti-CD36 in the body.
Okay, excellent. I guess that was the last one.
Okay, so I think we will thank you so much, Dr. Ho, for this fantastic talk, really.
Thank you very much.
We're very happy that you were able to share your results with us. Thank you very much.
Thank you.
And thank you, Luciana, Cynthia, and Mercedes, for the excellent organization of this webinar and all the participants. And well, we hope we have you someday in person here in Argentina. And we invite everybody for the next webinar. I don't know if Luciana, you want to...
the screen. Can you see the screen? Can you?
Yes.
Okay.
Yeah.
So next webinar, also on the topic of immune metabolism, will be in 15 days, the 24th of June. So please pay attention because the time is different. It will be at 6 p.m. Argentinian time, and you are all invited to participate of this webinar. It will be held by Dr. Edward Pierce. So I don't know whether you have anything else to add. Any of you? No? Okay.
Okay.
Yeah, I know.
So,
and I know Ed. I know Ed.
No.
I know him quite well.
Thank you. Yes, I can imagine. Thank you again on behalf of the Argentinian Society of Immunology. And okay, well, I think that's the end.
Thank you, Dr.
Thank you very much.
Have a nice day, everybody.
Okay, stay safe, stay well. Bye-bye.
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 Bye. Thank you very much.